All right. Hi, everyone. Let's go ahead and get started in the interest of time. I see a few more people still coming in. Thank you all for joining our PACVEC seminar series. Today is the next in our annual series during the academic year, and then we'll have one more seminar to close out the series in early June. So today's seminar is another very exciting one to our center because it features some of our recipients of training grants, and the two speakers today will be presenting their projects that were funded in the first round of training grants in 2018, 2019. So these at least started to wrap up last year. And first we have Josh Arnbrister. Josh is at the University of Arizona. He received his Bachelor of Science in Zoology in 2016 from Oregon State University. Then he took on a couple of research technician positions, one of which was at Oregon State. And then after that, I think in what might have been a formative experience for his career, he took a job as a field technician at Clackamas County Vector Control District, where he had a wide range of field experiences. He participated in testing local mosquitoes for Wolbachia, as well as various pesticide bioassays. And so a lot of those skills are certainly very relevant to public health in our region right now. And Josh is now pursuing his master's degree with Dr. Casey Ernst's lab at the University of Arizona. And as you may know, Dr. Ernst's lab and Josh are very focused on community engagement. So they have a Cadenga app for dengue and Zika surveillance along the US-Mexico border. And today, Josh's talk is entitled Engaging Community Health Workers in Mosquito Surveillance in an Urban Desert Environment. So with that, Josh, feel free to take over sharing your screen. And if you have any issues, let me know. Just make sure you unmute yourself.
Okay, will do. All right. Hey, everyone. I'm Josh. Thanks for the introduction, Chris. So I'm going to be talking about a project we did over the 2018 and 2019 mosquito season here in Southern Arizona called Engaging Community Health Workers in Mosquito Control and Surveillance in an Urban Desert Environment. So for this presentation, I'm going to cover a couple things. Going to start by giving some background on community health workers, and then about the Aedes aegypti situation and how control and surveillance is done in Arizona. And then I'm going to talk about the project itself, which we have since wrapped up, and then talk about kind of the results and conclusions we drew from this project. So starting off, what is a community health worker? So the definition we used is that a community health worker is a lay member of the community who works either for pay or as a volunteer in association with the local healthcare system. And they can work in both urban and rural environments, and are usually connected to the communities they work in by a shared ethnicity, language, or socioeconomic status. And so they really fulfilled this role of providing healthcare in areas that are sometimes missed by traditional healthcare methods, and in communities that are sometimes looked over. So as of 2018, there were 56,000 community health workers in the United States. Most community health workers in the US are female, 82%. And most of the work they do is targeting chronic illness. So things like heart disease, diabetes, some other things like vaccination, pregnancy health. So they're not necessarily focused on infectious disease, but they are very focused on different healthcare issues in their communities. And they work very closely with their communities. So that's part of the reason that we felt they would be ideally situated to perform tasks involving mosquito-borne illness and infectious diseases. So in Arizona, where we are, there is a really strong community health worker presence. In 2001, the Arizona Community Health Worker Association was founded. And this is just kind of an organization that provides professional development as well as a policy voice for the community health workers in the state. As of 2015, there were 1,000 community health workers in the state, in all the counties and all the tribes in the state. So we have this huge network of community health workers across the state, and we wanted to see about using them as part of mosquito surveillance. So the mosquito we specifically wanted to target was Aedes aegypti, which is pretty well established in the Arizona-Sonora region. And this is important because while we don't have any local transmission of dengue or Zika in Arizona, the Mexican state south of us, Sonora, has a lot of dengue and Zika transmission. In fact, in 2018, they were the highest state in Mexico for Zika transmission. And in Arizona in 2014, we did have a local outbreak of travel-related dengue cases in Yuma, which is right over here in this cluster. So how does Arizona deal with the Aedes aegypti threat? So most of the mosquito surveillance and control is done by local health departments, which isn't ideal because they have to deal with every other health issue that's out there. So in this case, so like for a recent example, they have to deal with things like the coronavirus outbreak, which takes away from funding and resources they could normally use for mosquito control and surveillance. We do have 2 local mosquito control districts in Arizona. One of these is in Yuma, and one of them is in Maricopa County. So these are great because they do only focus on mosquito-borne disease. But there are some limitations in terms of how well they can treat Aedes aegypti due to their ecology and habitat. And this kind of comes out in the way they control for them. So the main ways that these vector control districts are doing vector control is larviciding dry wells and storm drains and adulticiding. So dry wells or storm drains, like the one on the right here, are not ideal Aedes aegypti breeding habitat. Aedes aegypti is a container breeder, so they don't generally breed in these large bodies of water. So as such, these treatments are not really affecting them. And then some work done by Dr. Kathleen Walker, Dr. Ernst, and Dr. Mike Reilly at the U of A have shown that the adulticide is also not having a significant impact on Aedes aegypti adult density. So our hope was that by engaging another party in this control and surveillance that has more access to cryptic breeding sites, to people's houses, to areas of the community that vector control might not necessarily be able to reach, we could provide another way to battle Aedes aegypti. So this brings us to our current project, which is wondering, can community health workers be engaged in routine mosquito control and surveillance? So that's our hypothesis, was that community health workers can in fact be trained to identify mosquito larvae in habitat with a high success rate and can really help fill some of our current gaps in mosquito surveillance here in southern Arizona. So in order to test that, we split it into 2 parts. We wanted to test an interactive training module for community health workers, and then test the ability of community health workers to identify mosquito larvae. So in 2018, we launched a pilot project with a community health worker organization in Tucson called SERI, which stands for the Sonoran Environmental Research Institute. So this is a nonprofit organization that really focuses on environmental health in low-income communities. And what that looks like is they provide home inspections and look for things like missing fire detector or smoke detectors, seeing if smoke detectors work. They help set up water harvesting operations for people to use to water gardens and such. They check for screens, do lead testing. So they're kind of ideally situated to do the tasks we were thinking of. They're going to people's houses. They have access to yards, so they can look around for mosquito breeding sites, look for mosquito larvae, and record those larvae. So we started by doing a didactic classroom training with the community health workers from CERI we were working with, where we covered some basic things about mosquito life cycle, the Arizona mosquito species, how mosquito-borne disease works, what some of the symptoms are of different diseases. And then we really got into where mosquitoes breed, how to find mosquito breeding sites, and what type of preventative measures can be taken to prevent mosquitoes from breeding. And then we follow that up by doing a field training. So this was a hands-on demonstration of mosquito larval surveillance and habitat identification, and we did some guided inspections of volunteer residences. So a lot of the places that we did were community health workers' houses where they volunteered to have us come out and look for mosquito larvae at their house. And so we would identify breeding sites with them and then see if we found any larvae. And we always did. And they were very, very good at finding these larvae, to be honest. They found many larval habitats that I missed as the supposed expert, which was great for me that they were doing so well. So then after we did these trainings, we asked them for the rest of the mosquito season, which is about July to October, to continue collecting samples during their routine housing inspections. So whenever they found a mosquito larva at a house, they would collect the larva, put it in a little vial, and send it to us at the U of A. And then we would identify the larva, the species. We'd also take down some geographic data of where the larva was from and the types of habitats they were from. So the results of this first season was that the community health workers were very successful at identifying cryptic mosquito breeding sites, mosquito larvae, and Aedes aegypti adults. We had a lot of Aedes aegypti adults flying around at a lot of these sites, and they got very good at just identifying them by seeing them. Then we had a 1 month trial, or our trial period, where the community health workers delivered samples, and we got a mix of Aedes and Culex mosquitoes. There were 7 samples overall. Only one of these samples was not a mosquito larva, it was a fly larva, which is really my fault because I didn't go over aquatic flies in the training, so they weren't really clear on the difference. So after this first season, we had a focus group with the CERI community health workers to figure out what went well this season, what didn't go well, and how we could improve it for next season. So the first issue was we had this paper survey that we had them filling out whenever they did a housing inspection to kind of know where the house was, what type of breeding habitats they were finding, and if there were mosquito larvae. And the paper survey was really long. It was 2 or 3 pages and just took quite a while to fill out, which plays into the next issue that larval surveys were just taking a lot of time. So they were taking about 30 minutes, which is usually how long a normal housing inspection takes for them. So it was doubling the length of time they'd have to spend at houses. So we wanted to find a way to streamline the survey so that that would take less time. And then CERI was looking at making the larval survey separate from their housing surveys so that they could do them more efficiently. And then the other thing that they asked us for was a lot more regionally specific and detailed educational materials. They were really, really interested in kind of the nitty-gritty of mosquito-borne illness and symptoms and what that looked like. And also like really regional information on mosquitoes, like Southern Arizona, Tucson specific information. So then in our second season, we continued working with Siri and trying to address some of these issues that we had in the first season. And we also partnered with another community health worker group in Yuma called the Regional Center for Border Health. This is a very different organization than Siri. They are a walk-in clinic that focuses on providing healthcare and health programs to low-income communities. So they are a more traditional community health worker organization in the sense that their focus is on a lot of chronic diseases such as heart disease and diabetes. But they were interested in trying to add this mosquito-borne disease and mosquito surveillance component to their program. So the trainings were basically the same from the first season, but we added a lot more information on mosquito-borne disease. And also this time we developed some pre and post surveys on the trainings to see if the didactic training was actually working, which we hadn't done the first season. And then we continued to provide them with larval surveillance supplies and surveys to give at the houses. So to make the surveys more efficient, Siri did develop an electronic version because they do all their work off tablets. And that was done to make it easier and make these inspections take less time. So the pre and post surveys were scored out of 47 possible points, and we did give these surveys in both English and Spanish, since many of the community health workers speak Spanish as their primary language. And the pre and post surveys were completely identical. So it was really just seeing, like, did their score improve from the pre-survey to the post-survey. All the questions were multiple choice and matching and covered a wide range of subjects such as disease symptoms, identifying breeding sites, and mosquito control methods. So this picture on the right would be an example of one of our questions. We asked them to identify all the potential mosquito breeding sites in this picture and would then have them circle them and would end up with something like this. And then we would count how many sites they got and score that out of how many potential sites there were. So the results of this pre and post survey was that their mosquito knowledge did increase significantly after the didactic training. So we had 17 participants, and the post-survey scores were quite a bit higher than the pre-survey scores. And this was great. This is what we were looking for, that we were actually giving them some useful information and something that they were remembering out of these didactic trainings. So at this point in the study, we had indications that the didactic training model combined with hands-on training was very effective for teaching community health workers about mosquito-borne disease and mosquito surveillance. And we had these 2018 season samples that indicated that community health workers can in fact perform larval surveillance and can do it fairly well. However, we did have some limitations that showed up in the 2019 season. So in the 2019 season, we did not receive any larval samples. And the reasons for this did not involve the community health workers' ability to find larvae. What we ran into was issues of funding and workload. So on the right here, this image is from a study about how to build an effective community health worker program. And all of the circles on the outside are factors you need to consider. Things like effective training, things like is there a career path for community health workers? How the community looks at community health workers. And a lot of these things we had simply because Arizona has a good community health worker system. But the two areas we ran into issues with in the 2019 season were workload and compensation. So in terms of workload, the Regional Center for Border Health was not able to balance mosquito surveillance with other tasks they needed to complete. The mosquito season coincides with their busiest time at their walk-in clinic and when a lot of their community-based health programs start, because it's harvest season. It's also when school's out. So they're just doing a lot of things that aren't mosquito related, and they weren't able to do both. And then with the Sonoran Environmental Institute, we ran into funding issues, and they ran out of money on the grant that they had been using to do housing inspections. And so they ended up having to do other projects that season and weren't able to do any mosquito surveillance. And because of this, we didn't get any samples in 2019, which was a bummer. However, we did include in the survey a lot of questions asking if community health workers thought the trainings we were giving more important, if we thought mosquito disease and surveillance matched up with their job. And also we talked with a lot of them after these trainings, and the overwhelming feedback was they wanted this information about mosquito-borne disease, and they wanted these trainings about mosquito surveillance. And in all reality, it is really important that they have these trainings because most of the community health workers in Arizona are working in the Latinx community. And this community is disproportionately hit with the burden of dengue and Zika in Arizona. So on the right here, these are cases of dengue from 2008 to 2018 in Arizona by ethnicity, and 50% of them were in the Latinx community. And in a similar figure for Zika, you can see that 42% of them are in the Latinx community. So it's really disproportionate that how much they're hit by these diseases. And community health workers are often in the position to be the first people to see the symptoms of these diseases, to be the first people to see these diseases in their community. So it's really useful for them to have this knowledge, even if they're not able to perform mosquito surveillance. This stuff's really important to them. So overall, we concluded from our project that community health workers can perform mosquito larval surveillance, but there's a lot of logistical challenges that keep it from being practical for widespread use or something to combine into normal surveillance. That being said, we do feel it's really important to continue finding ways to bring mosquito-borne disease trainings to community health workers so that they can spread the information to their communities and other people that really need it. Yeah, so that was our project, and I'd like to just acknowledge my committee of Dr. Casey Ernst, Dr. Kathleen Wachter, and Dr. Don Goj for helping me out, as well as everyone in our labs, the community health worker organizations we worked with, and the PACVEC grant for helping fund a good portion of this project.
All right, thank you, Josh. Really nice presentation. Are there questions for Josh? Feel free to unmute yourself. You're all on mute, I believe. So if you have questions, just unmute, or you can type them into the chat.
Hi, Josh. Thanks. That was an interesting talk. I was wondering about the Zika and dengue cases. Were those autochthonous or imported cases?
Definitely imported. They're all travel-related cases, which is part of, the Latinx communities hit a lot harder since they're doing a lot more of the travel. But So yeah, they're not locally transmitted.
Thank you.
Josh, I was wondering, in addition to the challenges of time associated with the surveys, when you tried to train community health workers to identify larvae to species, that's pretty hard if you've got a lot of larval species to deal with. Did you just constrain the species such that got easier and didn't take so much time?
So we did actually train them to identify species and we didn't have any. I was doing the species identification. I gave them a lot of information of this is generally where 80s were found, so they could get an idea of which type, which genus it was. But we were gonna train them to find species, and then we decided that was just too much to ask. That'd be really hard. So.
Okay, I think this actually is analogous to the challenges in a lot of our region, and I'm thinking of the Pacific Islands particularly, where they also have environmental health workers who have a very broad range of duties, right. And mosquitoes and other vectors happen to be one of those. So I think funding or no funding, it's often hard to keep vectors in the view of people who have that many job duties. So it creates a lot of disparities in our region, where there are places that have focused people on vectors and then other people who are multi-purpose by definition.
Right, exactly. And that's part of why by the end of this, we were kind of thinking that it'd be more useful to provide the mosquito-borne illness training, and so that they kind of recognize diseases when they come across it, because they're very likely to do that, as opposed to doing full-on mosquito surveillance, which is significant.
Do you think doing this as an outbreak response would be more functional than having routine surveillance by community health workers?
Yeah. If you were responding to an outbreak, that community health workers would be a great resource to use during that, just because they do have such a close relationship with their community and a trust, so that it would be really easy for them to pick this up and really work with their communities.
Okay. Any other questions for Josh? All right. Well, thank you again, Josh. That was really great, and we really appreciate all the great work you've done on behalf of our center as well. With that, I think I see some claps there too. For those on Zoom, there's an option to clap virtually. So with that, we'll move on to Chase Ridenour. So Chase, if you want to go ahead and start sharing your screen, I'll briefly introduce you. Chase received his Bachelor of Science in Biology and Chemistry from Northern Arizona University in 2012. He then proceeded to do a 6-month internship in molecular biology in Indonesia. And I guess looking for some color in his portfolio, helped start a film production company back here in the US after the Indonesia experience, and a company called the Compass Initiative. So you can look that up if you're interested in some of those projects. I think one of their films focused on single mothers in Mexico, and is available on YouTube if you're interested. And then also after that, he joined a biochemistry lab at Mahidol University in Bangkok, Thailand. And then more recently has returned to pursue his PhD in bioinformatics, working with Dr. Crystal Hepp at Northern Arizona University. And that's the work he'll be telling us about today. Again, this is part of a training grant received from PACBEC. The title of his talk will be St. Louis encephalitis virus within 2 southwestern United States counties, Maricopa County, Arizona, and Riverside County, California. So take it away, Chase.
Thank you, Chris. Yeah, so I just wanted to give you a talk on the work that I did for the 2018 and 19 PACBEC grant that I received, and that we're finishing up this work in a paper that should be published hopefully in a preprint in the next couple of weeks, and will be published thereafter. So the work kind of the outline of the talk will be kind of a brief history of what St. Louis encephalitis virus is in the Americas, the 2 study sites and a sample acquisition, and then the main analysis where we did the phylogenetics of FLAB in the American Southwest, and looking at these 4 kind of major questions. So first, are all the genomes that we sequenced after the 2015 outbreak genotype 3? When did it arrive? What are the seasonal dynamics? And what is the migration pattern of it entering after it entered the US? And so first, kind of a brief history of St. Louis encephalitis. So St. Louis encephalitis is arbovirus. It is vectored by the Culex mosquitoes and is amplified in bird hosts, and it can be transferred into humans. Will be a dead end. So humans will be a dead end host. It cannot be spread after human, from human to human by mosquito, but the main amplification is from mosquito to bird and back, and then accidental transfer over to human.
Sit ups and leg lifts.
So if a person is infected, so 1% of infections develop clinical symptoms, and of these 1%, 40% of young adults and children, this is people under 50, will develop encephalitis. Then 90% of elderly, so this is considered 50 or above, will develop encephalitis. And then of the people who develop encephalitis, 5 to 15% of these, or it's a fatality rate of 5 to 15%. And currently there is no vaccine for St. Louis encephalitis. So it's a quick overview of the outbreaks. It's been in the Americas for hundreds of years, but it was first discovered in 1933 in St. Louis, Missouri, where it gets its name. And in that outbreak, there are over 1,000 cases and 201 deaths. And since then, there have been over 50 outbreaks in the United States and a couple in South America. And I'm just going to highlight a few of the larger ones since then. So Florida had 2 large ones in 1977, 1990, where they had 110, 226 cases respectively. Then the last major outbreak in the United States before 2015 was in Louisiana, that had only around 20 cases. And then this genotype 3 started causing outbreaks in Argentina, where they had 2 major outbreaks in 2005 and 2010, with 44 and 13 cases respectively. And then, so the 2001 Louisiana was the last outbreak until 2015 in Maricopa County, where Maricopa County had 23 cases, 19 neuroinvasive, or that had developed encephalitis, which resulted in 2 deaths. And the white dots are samples that I'll get into later. And so this is just a color map of the United States of cases after the 2015 outbreak. So the red counties are human cases, and the blue are just mosquito traps. But the main takeaway from this is that the majority of cases since the 2015 outbreak have been in the Southwest, and majority of those cases have happened within Maricopa County. And below, so then my 2 major areas, study areas for this are going to be Coachella Valley and Maricopa County, which These are two heat maps, which I'll go into individual later, but are just showing the seasonal outbreaks of SLE in each county. So going into more detail for each county, so we collaborate with Maricopa County Vector Control, and they do weekly sampling. They place 800 samples per week, and for a 12-hour period. And we will receive, and they collect them, test them for a bunch of different other like arboviruses, West Nile, Western equine, St. Louis encephalitis, like etc. If there's any positives, they'll send them to our lab, and then we process them. So, right. No. There you go. Sorry. The computer's having a little technical issue. Let's go forward. Sorry. Right. Okay, sorry. Now I have it under control. PowerPoint and my Mac don't get along very well. All right, so heat maps of, so what this is the black dots are the 800 sample sites that Maricopa Vector Control Agency does each week. And then the heat map is a yearly the density of positive mosquito pools in that area for that respective year. And main takeaway is that there's seasonal, but in 2016, it's the oddity, the positive mosquito pools found. But from 2017 through 2019 and continued in most likely into this year, there has been a consistent like high level of positive mosquito pools within Maricopa County. So our next study area is Coachella Valley Mosquito and Vector Control. We have a collaboration with them. Their trapping regimen is slightly different. They use 53 CO2 trap and gravid traps per week. These are, the CO2 traps are set up every 2 weeks, and they are also sampled at a 12-hour period. And for this analysis, we used 31 positive mosquito pools from the years 2017 and 18. And then these are heat maps. So you can see that the viral load in the mosquito populations is significantly lower in Coachella Valley. Like you can barely see it in 2017. And there's a couple of small hotspots in 2018, which I'll go into a little bit more detail on why we believe this is. So after we get these positive mosquito pools, we need to process them. And so we extract the DNA, the RNA viruses, so we reverse transcribe them to get them to DNA. And then we developed a multiplex PCR to sequenced the whole genome of S. levi in a single or in 2 separate PCRs. And kind of to give you an idea how these work is that you have the whole genome is about 11,000 base pairs, which is shown below. And so you'll have pool 1, which will be a set of primers, and they're spaced out so that the pool 1 primer will overlap with the pool 2 primer. And these are just different so that there's no conflict of the 2 primers running into each other. And then by having it spaced out, that they won't inhibit each other, and then we can sequence the whole genome. By doing it this way, it saves time because PCR reactions do not take as much time, and money overall. And so using this technique, it required 41 primer pairs. And we sequenced 137 positive mosquito pools in total, and we got an average of 95 to 99% coverage, which is high enough to continue with the phylogenetic analyses. And so after we sequence the results, then we have to process to get it ready for the phylogenetic analysis. So we trim off like the primer reads, line, and once we get the consensus sequence, then we can continue to the phylogenetic analyses, which we did a maximum likelihood analysis and a Bayesian analysis, which is what B stands for. So now looking into the phylogenetics. So again, just the questions that this analysis will answer are all the genotypes of all of the genotypes that we sequenced from Maricopa County and Coachella Valley of the same genotype of the 2015 outbreak. When did it arrive in the Southwest? What are the seasonal dynamics? And again, what are the migration patterns? So first, are all the genomes the same? We did this by doing a maximum likelihood tree, which we used all the public available genomes from both like North and South America. And those are shown in purple and gray. And then we also, the 137 genomes, which are in the blue and orange dots. You'll see an A over here. And it's hard to read, but these 2 dots here are the Argentina 2005 and 2010. And all of our sequences nest under, or nest within that and form a monophyletic clade under it, which is indicates that all the genomes are descendants of this 2015 outbreak strain, which corroborates earlier work and showing that it looks like it's just this genotype 3 circulating within these 2 counties. All right, so then the next 3 questions then will be answered using a beast analysis. And this stands for Bayesian evolutionary analysis sampling trees. And what this allows us to do is using a molecular clock, we can use the evolutionary relationships, but also then backdate like time of entry based upon these evolutionary relationships. So for this one, because we need to have accurate timing for the genomes, there are only 5 available public genomes that had specific dates. And then we used the 137 genomes from our lab, which was 106 from Maricopa County and 31 from Coachella Valley. The first question we're going to look to is, when did it arrive? And so this is the output of the beast tree, like unmodified. And the red branches here indicate branches that had low posterior. So meaning we have low confidence that this branching is real. So of like the 10,000 sampled trees, only, and like, so you'll see like some of these cases like 0 or like 15% of the trees had it, had this branching pattern. So we have low confidence in these branchings. And so what's not often done is we collapsed the branches that have these low posterior values. And what this allows us to see is the more true branching patterns instead of the like artificial branches with those low confidence. And so to answer the question of when did it arrive, we need to look at the root of the tree. The first, you'll see this naturally breaks up into 3 different clades. And the first clade is all the 2015 California and Arizona samples. And then clade 2 is majority is all the Arizona samples that we sequenced. And so looking into the date of arrival, I'll just zoom into clade 1. And you'll see the bottom is on like a time scale. And so if you look back at this will be the time of entry. So we estimated that the entrance into 2000 or into Maricopa or into the American Southwest was between September 2011 and May 2014, which had a median of 2013.5. And also with this clade, we can answer the question of, or try to answer, where did it arrive? Was it California or Arizona? But due to the like unclear branching, this polytomic like structure of this clade, we cannot confidently say whether or not it arrived in California or Arizona first. And it also indicates that there's high diversity. And so this also indicates that there were multiple introduction events over like a short period of time. So we're kind of hypothesizing like kind of like a multiple like infected birds within a single flock. It was like the introduction event. And so then going into the seasonality of SLEV within Maricopa County and Coachella Valley. To do this, we will need to look at because they split into independent clades, we'll inspect the structure of clade 2 and clade 3 to answer this question. So we'll see clade 2, majority is from California. There's only one exception, and it's not part of an extant cluster. But this overall clade splits into 2 temporal clades, or very monophyletic, and that almost all the 2017s cluster together and all the 2018s clustering together. And this is more indicative of a migratory event, or seasonal like migration event into Coachella Valley, rather than an endemic population seeding it each year, which is contrary to clade 3, which is the Arizona samples. As you can see, there isn't a clear clustering into temporal groups. There's like strong nesting, so there appear to be like multiple strains that have extant lineages in Maricopa County, which implies that there are endemic populations, and so there's seasonal reoccurrence by these local strains. And so then kind of going into what is the kind of larger like overall picture for the migration patterns of it entering. So this is the like hypothetical pattern. So for number 1, this would mean we entered into California. The introduction event would have gone into California and then entered into Arizona. 2 is the opposite, Arizona, and then migrated into California. And the next 2 are, number 3 is an uncertain origin and then split between Arizona and California. And then again, number 4 is unclear origin, but there's 2 distinct populations between Arizona and California. And so again, looking at the overall tree, it's difficult to tell because of the unclear branching in clade 1. So the origin between Arizona and California isn't clear, but there's distinct like California and Arizona, and then within those, there's the branching patterns as we discussed. So like the nesting within clade 3 showing endemic population or seasonal endemic circulation versus clade 2, which had the 2 distinct temporal clades, which is showing more like a migration event into it by like another local like California strain. And so right now it's difficult to determine which one, but because we have an unclear origin and some migration between the 2, I can't call which one exactly is right. I'm leaning, it appears more to be 4, so that Arizona and California are more 2 distinct clusters, but at the current point, we cannot rule out migration between the 2. So overall conclusion, that all the newly sequenced genomes are post-2015 outbreak genotype 3, corroborates previous work. That there was a multiple migration event of genotype 3 into the Americas, and this has occurred in 2013. And that there's seasonal migration events into the Coachella Valley and seasonal endemic circulation within Maricopa County. And with that, I would like to first thank PACVIC for funding a lot of the work, and then to all of our collaborators, especially Maricopa County, Coachella Valley, Arizona Department of Health Services, like TGen for sequencing, and then everyone in our lab, and NAU, and then Pathogen Microbiome Institute at NAU for allowing us to use their lab and make a lot of this possible. And with that, I'll take any questions. Thank you.